So, Handout 2H is going to talk about proteins. And proteins make up most of our body. So, when you think about most of the structure, that makes up things like bones, cartilage, uh, other connective tissues, structures that regulate our body, like hormones, enzymes, all of these are made of protein. And so, protein is a really important biomolecule. And all proteins are made of amino acids. And amino acids are the building blocks of a protein. And there's only 20 amino acids that make up all the proteins in your body. It's just the order of the amino acids that actually determine what protein we make. So the difference between a muscle protein and a protein that makes up collagen or a hormone protein is the arrangement of these amino acids. And I have written here that it says, check out handout 2K. And 2K shows us the 20 amino acids. And I'm going to pop them up here. I'm not going to have you memorize this chart. I won't have it on the test or anything. I just wanted to show you the different amino acids. So I wrote here that every protein is made out of these 20 amino acids. And you've probably heard of some like glycine or leucine, valine, tryptophan. Tryptophan is that amino acid that supposedly makes you sleepy at Thanksgiving because turkey's loaded with tryptophan, which is actually false. I mean, turkey does have tryptophan, but it doesn't have enough to make you sleepy. What makes you sleepy at Thanksgiving is overeating and having a bunch of blood rush to your digestive system. And then, of course, watching that hideous Dallas Cowboys football game, and it just puts you to sleep. So, proteins are made from these amino acids. Now, I'm not going to ask you specific questions about these particular amino acids. I just want you to know that amino acids make proteins. And what happens is they get bonded together by a covalent bond, and they're special bonds, so we call them peptide bonds. They build proteins. And of course, the protein structure is determined by the sequence of amino acids, like I just said. So, the difference between oxytocin and antidiuretic hormone is the sequence of proteins. Proteins have a structure, and it's kind of in this picture here. So if you look, it says we have a primary structure, which is just the string of amino acids. Those can be kind of coiled on each other to make a secondary structure. And when they fold to make a three-dimensional structure, that's called the tertiary structure. And then most proteins are more than one amino acid chain kind of stuck together. And so if we stick a bunch together, like in this picture, then we get what's called the quaternary structure of a protein. And that's usually the functional protein is made up of more than one amino acid chains all folded three-dimensionally on each other. Now, one of the perfect examples of a protein are enzymes. And enzymes are a type of protein that act as catalysts that lower our activation energy. So, when we are performing chemical reactions in our body, these chemical reactions would normally occur at extremely high temperatures. So imagine like we're going to break that disaccharide called sucrose into monosaccharides. So, just to give you an example here, let's say I've got a disaccharide and it's bonded together. This disaccharide's bonded together. If I want to break that bond, if I wanted to take sucrose and break it down into glucose and fructose, which are the two monosaccharides that build sucrose. If I did that in the lab, it would take hundreds of degrees. I would have to put it in a Bunsen burner, and we'd have to heat up that sucrose sugar to get it to break into glucose and fructose. Well, our body doesn't run at hundreds and hundreds of degrees, right? It runs at 98.6 degrees Fahrenheit or 37 degrees Celsius. That's what our body runs at. And so what an enzyme does is it lowers that activation energy that breaks the sucrose into glucose and fructose. It lowers that energy down to 98.6. And our enzymes all function at our body temperature. That's why our body temperature is our body temperature, because human enzymes work at 98.6 degrees Fahrenheit. And so if we get too far away from 98.6, really what puts us out of homeostasis, what causes our body to dysfunction, is that our enzymes start shutting down, because the enzymes want to function at that temperature. So, for example, we can't break sucrose into glucose and fructose if our body temperature is too far away from 98.6. And that's why having a really, really high fever or having a low temperature of hypothermia can cause death, because essentially our cells cannot perform all of their chemical reactions because the enzymes are shutting down. And so enzymes are able to lower our activation energy to 98.6. So some kind of chemical reaction that would require hundreds of degrees in the lab can occur with an enzyme at 98.6 degrees. Now, there are two kinds of chemical reactions. There are anabolic, which build, and we talked about this in a previous video, and there are catabolic that break down. So what enzymes do is they cause anabolic and catabolic chemical reactions. So if you look at this example here, we've got two molecules. When an enzyme grabs them, we can put them together. That's anabolic when we're building it. Or if we go the opposite direction, if we have one chemical and we add the enzyme to it, we can break it apart to make it catabolic. And so these enzymes are going to be extremely important in keeping our body in homeostasis, and they're made of proteins. That's what they do. So that's just one example of an important protein. So that concludes this handout, 2H, and we'll move on to our next biomolecule, lipids, for handout 2I. So until then, I'll see you later. Take care.